So this semester, I worked in my molecular biology class, taught by Dr. Janine O'Brien, on a research project, which had three main objectives. The first was to identify the presence or absence of 16S RNA genes in snow samples to determine if bacteria is present in our samples, to identify the presence or absence of INP genes, such as INAQ, in snow samples to determine if different nucleating bacteria is present. And then our third objective was to compare abundance across samples for 16S genes to determine if various species of bacteria are present. So a little background information. Snowflakes consist of water and small particles, which could be particles such as dust or bacteria. And a little fun fact is bacteria is everywhere. It's on the ground, it's in the air, it's up in the clouds. So if water vapor droplets condense on those small particles, it tends to freeze because of the cold atmosphere, and it can cause this branching to occur, such as indicated in figure A of figure 1. It shows that particles are present in the clouds, water vapor condenses onto those particles, and as more and more bind, it creates this growing branching, and it will fall down as snow, as many people are aware of here. So winter storms tend to be affected by the lake, such as Lake Superior has great lake effect on winter storms. And the lake water is usually warmer than the colder air above it. And once the warm and cold fronts meet, moisture is caused from the rising warm air, and this can help produce clouds. And then the snowflake formation helps aid in the winter storms. And as this occurs, as shown in figure B and C, storms tend to move west to east. And it is thought that different bacteria is present in different regions. So it's hypothesized that different storms, as they move from place to place, will carry different types of bacteria with it if those nucleating bacteria are present in the storm clouds. So we kind of want to identify if bacteria is present at all. We want to know if the nucleating bacteria is present, and then we want to compare the bacteria samples to identify how many and the identity of the different bacteria that could be in these different snow samples. So the snow samples that we used were collected from Dr. O'Brien's backyard, and we extracted the bacteria from the snow using an AeroPress with a filter paper on the bottom. So as snow was pushed through the AeroPress, any bacteria that could be present in the samples were collected on the filter paper. And this process is shown in figure 2A, where we have different potential bacteria on the snowflakes, and we used filtration to have a filter paper with just bacteria on it, which could have some small contaminants such as dirt on it as well. So we used a Q-Gin kit to kind of remove any other contaminants and lyse open the bacterial cells to extract the bacterial DNA that we want to analyze and identify in our experiment. Part B is just showing the steps that were taken with the QGen kit, which was typically used for stool, but because we used it because of its ability to use large sample sizes at a time and identify many different species of bacteria, which would also be present in stool. So once we purified and isolated the bacterial DNA, we used PCR to amplify our samples. And then we loaded those onto a 1% agarose gel and ran gel electrophoresis to visually analyze our results. The first round was used with 23AAP, 23AT, and 23AS samples. And we were specifically looking for the 16S gene, which would be indicated, its presence would be indicated by a band around 1 kb. And our results, shown in figure 3, show that there was no bands at 1 kb seen in any of our samples, which indicate that there was not any bacteria present in our samples. So from there, we've identified different samples that were known to have 16S bacteria from prior years. So we took those samples, 23BT, 22AT, and 22AP, and we wanted to identify whether or not we had those nucleating bacteria present. So specifically the INAQ. So we use INAQ specific cycles for our PCR. And we also loaded those onto a gel and ran gel electrophoresis. And our results show that no nucleating bacteria or INAQ were seen in our gel or our samples because of the absence of the bands around 3 kb, which is where the INAQ band would be present. So our conclusions were the 23AP, 23AT, and 23AS samples did not display the 16S gene. And when we moved on to the 23BT, 22AT, and 22AP samples, they were known to possess 16S gene, but they did not display a presence of the INAQ gene. Our objective 3, where we were not able to meet or implement in this experiment, but our approach to analyzing that would be using Illumina sequencing. And Illumina sequencing is very beneficial because it provides a lot of information, including the amino acid sequences of all the bacteria present in the sample, information about the abundance of each bacteria, and information about whether there is variation in that bacteria. So how that would work would be we would take our 16S genes and we would have that go through that method and sequencing, and it would tell us all the different types of bacteria that had 16S that were present, and we can individually not only see how much, like percentages of how much is in our sample, but also be able to identify the specific species present in our samples. So that would be our future steps, and we would want to pursue that.